that light. Okay, last lecture of quarter 2. Yay, urinary system. And again, urinary system is rather complex machinery, but we're going to break it down factoid by factoid, one piece at a time, until each and every piece makes sense and you know exactly what to focus on. There's not going to be a big mystery about what do I study in this enormous system. It's going to be I study this large amount of information in this system, and it's just going to be very specific. So we'll do general structure and functions of the urinary system. We'll do renal anatomy, the kidneys, the mechanism of urine formation, which is our big topic of the day. That's the nephron. You're going to be a big fan of the nephron by the end of the day, at least I hope so. How the kidneys regulate blood pressure. We've actually seen this a little bit. We saw it for endocrine, and we saw it a little bit for blood pressure, just when we were doing our blood system, our cardiovascular system. So that should be review. Renal clearance. It's going to be related to topics of pharmacology and how our renal system helps us to clear drugs and what that means. And the urinary tract, the lower urinary system, including the urinary bladder and urethra, which are relatively simple compared to everything else we're covering today. So anatomically, the urinary system, or the renal system, is going to be how the body filters blood to create urine, forms urine, which is a waste product, and how that drains to the urinary bladder and is voided and excreted. You already know that the kidneys are retroperitoneal, so there will be the peritoneal membrane covering that kidney. But then you remove that parietal peritoneum and you can get this view right here. And now you can see the kidneys are going to be located just under the respiratory diaphragm, mostly nestled and protected by those lowest ribs. And there is some significance to the fact that the right kidney will be slightly lower than the left kidney. What is there anatomically that would push that right kidney down a little bit? The liver. So they are relatively protected. Right kidney slightly less protected than the left because of that anatomy. You know about the adrenal glands. They are superior to the kidneys. So the kidneys themselves, the ureter right here down to the urinary bladder to the urethra is how that pathway works. The urinary system will store urine, excrete urine, regulate blood volume, both in short and long term, multiple mechanisms in which it participates to regulate blood volume. Regulates erythrocyte production. Does anybody remember the hormone that the kidney releases to initiate red blood cell production? I can see it's right on the tip of your tongues. You've got this. You know it. Something poiesis. Erythropoietin. Well done. Excellent job. Okay, so erythropoietin, so that's part of how it regulates red blood cell production. And then regulates ion balance and acid-base balance. When you get to patho, when you get to quarter 4, you're going to start caring a lot about ions and acid-base balances. It's going to be very key to the way you treat a number of illnesses. Again, those kidneys are retroperitoneal. There are renal arteries and renal veins that serve them. So they're very, very closely associated with that abdominal aorta and that inferior vena cava. That's what you're seeing right here. So this is a transverse cross-section at the level of the L2 vertebra. And you can see part of it, you can see the peritoneal membrane covering things. So you can see the liver. This is the liver over here. This is the stomach. These are bits of intestine, and there's that spleen over there. Now this is going to be what we do with our dissection today. It's just going to be a coronal section of the kidney, and you'll be able to see that within the kidneys you have some structures. You have a cortex. You actually have a fibrous capsule. It's not on your vocab list right now, but this is known as Gerada's fascia, and that will be significant to you one day. So the fact that it's encapsulated has significance. Then there's a cortex, and then there's a medulla, and the medulla is going to be made of pyramids that are shaped like pyramids and columns that are shaped like columns. And you should more or less be able to see them on your kidneys today. They may or may not be as well defined as this, depending on your individual specimen. The direction of filtration in general is going to be that it's going to go from the cortex towards the medulla, and within these pyramids, things are going to drain towards these structures known as the minor calyces. It's a minor calyx, singular. So urine drains to the minor calyx, and then to a slightly larger major calyx, and then to this space known as the renal pelvis. This will be the renal pelvis, and that drains directly to the ureter. Now the ureter does have some smooth muscle around it. It's also aided by gravity if you're upright. So urine flow should always be this direction overall. The blood supply to the kidneys is of great significance to you. This is a very vascular organ, and because it's a vascular organ, because there's a lot of microvasculature, it's going to be vulnerable to a number of diseases related to microvasculature, so you do want to learn the specific vascular supply. So it starts with the renal artery, and those are going to divide into segmental arteries. Each of those are going to further divide into interlobar arteries. So those interlobar arteries serve basically lobes of the kidney, which is to say they travel along the columns and serve the pyramids. So interlobar artery, and that's going to arc around that base of the pyramid out towards the cortex. And at that point we call it an arcuate artery. And that arcuate artery is going to have these little radiating arteries off of it, and those are known as interlobular arteries. So don't mix those up. This was interlobar, and this was an interlobular artery, and there was an arcuate artery that split those two things. Now we're going to zoom in when we get to that interlobular artery. We haven't even looked at the nephron yet. Don't worry about it too much. We're going to have a little filtration unit known as a nephron, and it's going to be served by an afferent arteriole that's going to create this little capillary tuft known as a glomerulus, which you will be able to pronounce by the end of the day. And rather than deoxygenating here, it's filtering here. It is not deoxygenating, it is not losing oxygen. So the blood vessel that leaves this capillary bed is an efferent arteriole. That efferent arteriole goes on to supply the rest of the nephron, so the rest of this filtration unit, and it's going to continue participating in the concentration of urine throughout this structure, and it is also going to deoxygenate at the same time. So by the end of the nephron, you will be in the venous side of the circulatory system. Now luckily from here on out it is symmetrical, so you had an interlobar artery. The next thing that you come back up to is an interlobular vein, then an arcuate vein, then an interlobar vein, segmental veins, and then the renal vein. So once you've learned one side, you know the other. The only fiddly bits are down here at the nephron level. So far, so good? Yeah. That is a great question. The difference between the renal corpuscle and the glomerulus is technically just the capillary bed, and the renal corpuscle includes the capsule, plus the capillary bed, the whole structure. So for many of you, you're not there yet worrying about renal corpuscles, and that's okay. You don't have to be worrying about renal corpuscles just yet. The kidney is innervated via the renal plexus, and then pain from the kidneys usually refers from dermatomes T10 to T12. Do you guys remember where T10 dermatome is? For reference, it's a significant one. It's one of the major ones. Do we need to watch the embarrassing dermatome rap again? Well, the embarrassing phrase that the dermatome rap used for that is that T10 circles belly button hair. So that was your belly button. Yeah. Okay, two types of nephrons, cortical and juxtamedullary. And those are just ways of saying more of this filtration unit is located within the cortex, versus juxtamedullary means this will be right next to the adrenal medulla, and for more of it dips into the adrenal medulla. Nephrons have a structure known as the nephron loop. The old school term for this is loop of Henle, nephron loop of Henle. The cortical nephrons, the nephron loop does dip into the medulla, into the pyramid, but only a little bit. Whereas juxtamedullary nephrons dip very far into that renal pyramid, into that medulla. There's a reason for that. The more nephron loop is located within the adrenal medulla, the more we can retain water. So for humans, actually most of ours are up here. 85% of ours are cortical nephrons. That means we can retain some water, we're pretty decent at it, but we're not as good as camels are. If you're a camel, you're going to have mostly juxtamedullary nephrons. This is a structure that enables us to retain water, that enables us to concentrate urine so that we can get rid of wastes without losing a whole bunch of water. Big picture, that's what we're getting at today. If you remember nothing else about the renal system, remember that it's about getting rid of wastes while keeping ourselves well hydrated. Evolutionarily, ever since we came out of the water, our bodies have had to evolve to preserve water. Dehydration is the biggest threat once we became terrestrial animals. So we developed the renal system in response to this. Fish don't really have renal systems like this. They're in the water, they don't really need to worry about hydration. They have waste systems, sure, but not the same sort of renal system like we do. Whereas a camel has a highly evolved renal system. All right, let's start with an overview, and then we're going to hit this point by point by point by point. And I promise we're going to go over this 5 times in many, many different ways. This is the nephron. This is our unit of filtration and concentration and production of urine. That's going to happen across this. This is taking a 3-dimensional object and spreading it out in a linear fashion. So real life, they're convoluted. There's even the word convoluted in the name. They're going to wrap around in 3 dimensions and come in contact with various parts. But we're just straightening that out so that we can think of it in a linear fashion. And the way it's written on your worksheet is pretty linear as well. So first things first, the afferent arteriole brings blood into the renal corpuscle. And thanks to things like hydrostatic pressure, your really good friend hydrostatic pressure, fluid is pushed out of the glomerulus and into the capsular space of this structure known as the renal corpuscle. So your glomerulus, your renal corpuscle, is the basic unit of filtration. This is where bulk filtration occurs. This is the main site of filtering. We're going to lose a whole bunch of stuff from our bloodstream into the capsular space, and at that point it's going to be known as filtrate. The fluid located right here is known as filtrate.
Located in the, what did you say, the corpuscle?
Capsular space. Yep. Filtrate is within the capsular space. That filtrate is going to continue along this tubule system, and by the time it gets to here, it's going to be known as urine. That urine is only going to be about 1% of what was filtered here. So you're actually going to reabsorb 99% of the fluid that was filtered between here and here. All right, so we've got to filtrate. And that filtrate is going to contain fluid, water, ions, proteins, sugars, waste products, nitrogenous wastes, a number of other things, but those are going to be our focus for the day. If you're not scribbling, you're not looking at your worksheet right now.
Fluid, water, ions, protein, sugars.
What's that?
I couldn't keep up.
I think we've only answered 2 of them so far. Maybe 1.
No, what? The first 3.
The first 3? That's good. Okay. All right, that's okay. We've got the rest of the day for this. And you're going to keep hearing those phrases again and again. Ions, proteins, wastes, glucose, drugs, things like that. We're going to hear that over and over again. That's going to be repeated. That's going to be a theme. Yes?
Did you say it was hydrostatic pressure that pushed it into the
Hydrostatic pressure out. And this is the same thing. This is a capillary bed. So hydrostatic pressure and osmotic pressure are both pressures that act in this space as well. But yes, the hydrostatic is the same old push that you are well familiar with at this point. Okay, the next stop is going to be the proximal convoluted tubule, also known as the PCT. And yes, you are allowed to call it the PCT. You should be able to recognize proximal convoluted tubule. You should know what that means. I think it's beneficial for understanding its location and function. Now, there's a huge concept for this entire system, and those are the ideas of reabsorption and secretion. In this system, those are one-way terms. You have the tubule system, tubes, and you have the cardiovascular system. Reabsorption refers to fluid moving from the tubule system to the cardiovascular system. Reabsorbed.
Could you say that again?
Reabsorption refers to something moving from the tubule system, this nephron here, back to the cardiovascular system.
So moving from the nephron to the cardiovascular system?
Mm-hmm. Correct. Secretion is the opposite. It means moving from the cardiovascular system to the tubule system. These are one-way terms, very, very explicitly one-way terms. One thing you never ever want to say is secreted to the cardiovascular system. You never ever want to say reabsorbed to the tubule system. Those are incorrect for this system. And part of that is there can be an exam question, then it will just use the word reabsorb or just use the word secrete. This has happened in the past with actually my metro class, not necessarily here, where somebody said, what do you mean by reabsorbed? And I had to say, that's part of the question. I can't define that for you. So these are one-way terms. So when you see these arrows, you see this arrow specifically meaning reabsorption and this arrow specifically meaning secretion. Because if it's secreted, it's going to be excreted. If it's secreted to the tubule system, its destiny most likely is to leave the body as urine. Whereas it's reabsorbed by the body, the cardiovascular system. It stays within the body. So does it make sense why these are only appropriate in one direction? So secretion will end up secreted or excreted as urine, whereas reabsorbed will remain in the body via the cardiovascular system.
Okay.
Yep. And again, we will go over this piece by piece again, so if you don't catch everything right away, that's okay. At the proximal convoluted tubule, we start the process of reabsorption. We're going to reabsorb some water, some sodium, some ions aside from sodium. We're going to reabsorb some glucose. And we're actually going to secrete many drugs here.
Is
that where most of the reabsorption occurs?
I would say so. Most reabsorption is going to happen at the proximal convoluted tubule. In fact, you're going to have microvilli, dense microvilli along here. Why do we ever have microvilli? That's cilia.
Oh.
Surface area.
Surface area. Microvilli is for surface area. So the PCT has dense microvilli to increase surface area so that, yes, most of our reabsorption can occur here. For most of our ions, for a lot of nutrients, we will reabsorb most of that here, and we will secrete drugs in the PCT. Then we move on to the nephron loop. And again, there's more slides for this coming up. And the nephron loop has two parts, the descending limb and the ascending limb. Sometimes we refer to the descending limb as thin limb and the ascending limb as thick limb because of the thickness of the walls. This will be simple cuboidal, this will be simple squamous down here. What this is going to do is in two parts, and part one actually happens further down the road. In the ascending limb, we're going to pump out sodium and chloride in the ascending limb. Now, if you're water, where do you want to go? Towards stuff. Is sodium chloride stuff? Sodium chloride is absolutely stuff, especially sodium. Let's focus more on sodium rather than chloride. Now, the descending limb is permeable to water. So if you have pumped out a whole bunch of sodium on this side, and you're a whole bunch of water in a permeable environment, where are you going to go? It's going to go out to technically the adrenal, or not adrenal, sorry, the medulla, the renal medulla, and then back to the cardiovascular system. So it goes through those compartments, through the medulla to the cardiovascular system. So you're going to have active pumping of sodium in the ascending limb, and you're going to have passive osmosis of water in the descending limb. Water is going to be reabsorbed in the descending limb, and sodium and chloride are going to be actively pumped out of the ascending limb. Big picture, what you're doing here is first of all setting up a saltiness gradient. We're going to get salty like League of Legends out here. No laugh. One person's laughed at that so far. Yeah, okay. We got it. Somebody amused. That's enough for me. I'm going to keep using it. It's never going to end. Okay. We got some saltiness going on here. What else is salty? What's like a more applicable to the general population use of the word salty for you guys?
Well, it depends on how people take salty.
Yeah, like the modern usage of like, wow, she's salty. She's feeling salty today. No? Nobody uses that? Just me? Okay.
Yeah, I used it not too long ago.
I used it.
John Lennon.
About who?
John Lennon.
John Lennon is salty. Okay.
And I like that about him.
That's good. I'll find one. I'll find a more socially applicable one. So we're setting up a salt gradient is really what we're doing here. We're going to make it salty, salty, or saltiest as we get further and further into that renal medulla. And that's going to mean that anytime water can be reabsorbed, water's going to want to reabsorb. Water's going to want to go towards that salt. So it also means that over the course of this nephron loop, we're going to further concentrate urine. I mean, big picture, that's all we're getting at here, concentrated urine. We go from a lot of fluid and a lot of salt here, a lot of water, a lot of salt, to less water and less salt. So by the time you get through this nephron loop, you've retained a lot more of just water and salt here. Sound good to everyone? Okay, let's move on to the DCT, and again, we will be repeating this on future slides. There's more detail to come. I'm just making sure we've got an overview for now. The DCT is going to be pretty similar to the PCT, but we no longer have those thick microvilli, so we don't have as much reabsorption surface area. So we will be reabsorbing some of the very similar things. We will be reabsorbing more water, especially because we'll be reabsorbing more sodium, more ions, more glucose, just everything that we've been talking about so far, really. Now, one thing I want you to focus on for the DCT is that it's going to respond to our friend, the hormone aldosterone. Aldosterone, you already know about. What do you know about aldosterone?
As part of the RAS.
Perfect. It's part of RAAS, renin-angiotensin aldosterone system. What does it cause?
Blood pressure.
Increased blood pressure. But due to what? Increased what?
Water retention.
Blood volume. Water retention, blood volume. Absolutely. So what aldosterone is going to do is it's directly going to cause sodium reabsorption, but potassium excretion, or secretion, I should say. That's going to help us differentiate it from ADH, which is going to work over here. And it's also going to affect ion balances. And I mentioned earlier that when you get to patho, when you get to quarter 4, high acuity nursing, you're going to care a lot about ion balances. You're going to be tracking people's sodium and potassium. So if this thing causes sodium retention, sodium reabsorption, and sodium secretion, or sorry, potassium secretion, you're going to care about that. Do you agree with that? Yeah. What's that? Do you ever do anything with ions at all?
No.
No? Not in emergency medicine then? That's good. Okay. All right. So sodium retention, potassium excretion, and because you retain sodium, sodium's going this way. Where's water want to go? With it. Water goes too. Okie doke. That's the DCT. Now we've gotten to the collecting duct, or collecting duct. Now the collecting duct is shared. There will be multiple nephrons going onto one collecting duct. And it's mostly going to be a passageway. This is going to lead to those minor calyces, major calyces, renal pelvis, and ureter. By this point, you can safely call that urine. But there's one last opportunity for your kidneys to retain water. The cells of the collecting duct are going to respond to antidiuretic hormone, ADH. What released ADH? It is the pituitary. Well done. Posterior pituitary releases ADH. And the cells of the collecting duct will respond to it by inserting aquaporins into their plasma membranes. Do you guys remember what aquaporins are? We talked about perforin molecules. They're very similar. It's kind of in the name, aquaporin. Like a pore for aqua of some kind. It's literally, that's what it is. So it's a channel that allows water through. Now it's just a passive thing. We've got a little channel and it's open now. But over here at the ascending limb, what were we doing in the ascending loop of Henle?
Descending is secreting NaCl.
Yep, pumping out sodium. And what did we get out of that? Well,
Concentration.
we got concentration from the nephron loop. What did we get in the medulla?
Water reabsorption.
Water reabsorption because of what? League of Legends. Salt gradient. There we go. Saltiness. So we got a lot of salt out here already, thanks to that loop of Henle. And now, really close by, we've got a bunch of water channels. So what happens when we put a bunch of water channels in the collecting duct cells?
The water is reabsorbed.
Water is reabsorbed. So this is your last chance to reabsorb water. This is your last chance to stay hydrated. So antidiuretic hormone prevents you from peeing large amounts of water by allowing you to reabsorb that water at the collecting ducts. Therefore, you retain more water. That means that when you are dehydrated, your body will release antidiuretic hormone to maintain your hydration level. But when you are very well hydrated, you will stop secreting antidiuretic hormone. Those cells will remove the aquaporins from their plasma membranes, and the collecting duct will become impermeable to water, meaning that you will urinate a larger volume of more dilute urine.
Would that be in negative feedback loop?
Yeah, negative feedback loop. Definitely a standard homeostatic mechanism, negative feedback. All right, that was it. That was the tough stuff. We will go backwards and forwards over that. There are slides coming up to review this concept. So again, urine is less than 1% of the volume of the original filtrate. Filtrate is the definition of the fluid in the capsular space that was just pushed out of the glomerulus. Let's zoom in on that renal corpuscle, and shortly after this we're going to take our first break. There's the afferent arteriole, there's the glomerulus, there's the efferent arteriole. Within the renal corpuscle you have a visceral and parietal layer. Just like you do in any space in your body, there's a visceral and parietal layer. Now the parietal layer is the layer adhered to the renal corpuscle, and that is a simple squamous epithelium. And that's all you got to know about that. But if you look at the capillary bed here, there's another type of cell here known as a podocyte. Those podocyte cells are going to surround the capillary bed, and they have little feet, little foot processes known as pedicles or pedicels, depending on who you ask. And those foot processes are going to go around and sort of join up with each other and help create the filtration membrane. Which part? Podocyte, pedicel? I think I should have it somewhere on here. Here we go. Podocyte, pedicels, right there.
So the podocyte is the big one and the pedicel is the feet.
Yeah, so the podocyte is the entire cell,
Okay.
and those pedicels is the entire cell. Those foot processes are the pedicels. They are part of the cell. They're cellular extensions. So they join together to help create the filtration membrane. The other part of the filtration membrane is that the capillary bed here that makes up the glomerulus has fenestrations. This is a fenestrated capillary bed. So what do fenestrations allow through versus what do they maintain?
They allow passive, diffusion of molecules.
So some diffusion of molecules is going to happen fairly well through this. So here's your capillary, here's your fenestration. There's actually going to be a shared basement membrane here, much like you had in your respiratory system, shared basement membrane between these spaces. And here are the pedicels and the filtration slits between them. So this is going to create a selective barrier. It's going to let some things out, and it's going to maintain some things. What things do we really want to keep in the bloodstream that it's a really bad sign if they're getting peed out? What do we not want to see in pee? Blood. We definitely don't want red blood cells. Not a large amount anyway. So red blood cells are going to be too big to go through the filtration membrane. What else is a bad sign if it shows up in blood, or sorry, in urine? Large amounts of proteins. Proteinuria is a bad thing. So though we will have proteins smaller than 3 nanometers, that shared basement membrane is going to make it harder for that to move through. So fluid can get through, ions can get through, very small molecules, urea, nitrogenous wastes, the things that make urine smell the way urine does, and the purpose of urine is to get rid of nitrogenous wastes, those will get through just fine. But larger proteins will be selectively withheld, whole blood cells will be selectively withheld, and if those things end up in urine, that means this membrane is compromised. Okay, I think that's where I want to stop before we get to the PCT before your break.